into the endoplasmic reticulum. And these little dots, but then you have, let's do some cytoplasm. So cytoplasm is a jelly-like substance that's inside of the cell. So you have the cytoplasm, and so this is the gel. This is what separates the inside of the cell from the outside of the world. So you have the cytoplasm. Then you have the nucleus, which is the blood. So we'll put that red because that's where the blood is at. And chromosomes. We got the DNA. So now, then we got the nucleolus that's inside of the nucleus that produces the ribosomes.
Now I have a question. The other day we were talking about cells, and Ari said, "What happens when there's too much waste inside of a cell?"
Now this is where the lysosome. Now this is a lysosome. A lysosome, we'll just draw it like a garbage truck or something. We'll just draw, that's the lysosome. So the lysosome is going to get rid of the waste that's inside of the cell. All of the stuff that the cell don't need. You got the garbage disposals, which is the lysosomes that cleans up the cell. And then you have a mitochondria, which gives it the power, the energy.
Mitochondria. That sounds like a funny word. Memnon, can you say mitochondria?
Mitochondria.
Mitochondria.
I be messing up the words. You gotta, cause I'm not educated. I dropped out in the 8th grade, so I don't know.
Well, you sound very educated.
So, the mitochondria, it's the powerhouse. It's what fuels the cell. It gives the cell its energy. You know what I'm saying? And its function to be able to reproduce. So then the ribosomes that's produced inside of the nucleus, it produces these proteins and these little dots. They are the ribosomes. So you have the ribosomes, and then you still have these like strands of DNA particles. You have your mitochondria. You have the Golgi apparatus. But when the lysosomes leave the ribosomes leaves the nucleus, they are in the vesicle. There's something like a vesicle or something. So if you still get these little dots of you got these little dots, these ribosomes. You have the ER, a rough ER and a smooth ER. But the ribosomes they're not in the smooth ER. They are being they get the proteins, and then the proteins the vesicles, they go out of the endoplasmic reticulum, and they have these vesicles. So that they go into that, pretty much twists up the DNA, add proteins and carbohydrates and things like that. So they come out of the nucleus and to the endoplasmic reticulum, and then the Golgi apparatus. The Golgi apparatus takes these proteins. So like we'll just say the Golgi apparatus.
You need more colors?
I just need something different to represent.
All right. Do you have any more colors over there, that grandpa? Okay.
Golgi apparatus. We'll say this is the Golgi apparatus.
Super duper color.
So this is, it takes these ribosomes, into the Golgi apparatus to be able to twist it and to add to things that's needed to be able to reproduce a cell. So you have and like the garbage disposal, something to clean the cells from the waste. You have the blood, which is in the nucleus, and then the nucleus is like a man and a woman. You have the nucleus, but inside the nucleus you have the nucleolus, and that's what produces the ribosomes, the proteins. So it's not just the nucleus, it's the nucleolus inside of the nucleus. that produces the ribosomes that enter into the endoplasmic reticulum. That you know, because the DNA, the deoxyribonucleic acid, is it has this genetic code called chromatides. You know, and the chromatides are twisted strands of blood, and it has a pattern like AG, this, and it just have different patterns that it's hard to be duplicated, and it's nothing like it. That's where we get our fingerprints from. This uniqueness of these chromatides that that separate us, but also links us to our host, you know, and our host is the person who our parents, who we call our parents, that had these sperm cells, you know, with these little heads, and then they would enter into, you know, the vaginal system, and you know, and then they would start reproducing, you know, and these, you would take the blood from these sperm cells, and then it would have all of the information within the nucleus and the nucleolus. And so you would take that, and then it would start producing